Hello everyone, my name is Mohammed Usman and I'm here to present the findings of my research titled AFP-CKSAAP Prediction of Antifreeze Proteins Using Composition of K-Spaced Amino Acid Pairs with Deep Neural Network. The paper is written by me under the supervision of my professor, Dr. E. Jionga. Before beginning the presentation, I would like to give you a brief introduction about what antifreeze proteins are. Well, it all started in 1950s when Dr. Backus from the Cornell University asked a simple question from his senior colleague, Dr. Sholender. He asked that in Arctic, when the fishes swim in the extreme cold temperature at -1.7 degrees, why do not they freeze? Well, this simple question resulted in a series of research that is still in progress. Dr. Sholender investigated and found some marine fishes living in the high north had a compound in their blood. This compound helped to lower the freezing point of the whole fish, and he called that substance as antifreeze. The true nature of this antifreeze compound was identified as glycoproteins by Dr. DeVries in 1970s. Well, let's take a look at the presentation flow. I will be giving you a brief introduction about how antifreeze proteins work. Then we will have a look about some of the antifreeze proteins type. This will be followed by the motivation of the research, and then I will give you a brief description about the contemporary work that has been done in this regard. Then we will take a detailed look on the contribution of this research, followed by the results and comparison. Then finally, the presentation will be concluded. The antifreeze proteins are responsible for the phenomena of thermal hysteresis. In this phenomena, the freezing point of the water is lowered to a few degrees without any alteration in the melting point. Also, the ice has an unusual property of recrystallization, in which small ice crystals grow to become large ice lattice and cause physical damage to the cell membrane and tissues of the organisms. The ice can exist in several polymorphic structures. Out of these, only hexagonal structure can be stable under normal pressure at 0 degrees. AFP contact sites bind to these ice lattice and interfere their growth along the prism plane, and makes it unfavorable for the water molecules to join the surface. Hence, they hinder the growth of ice lattice into a larger one. There are several types of antifreeze proteins that are found and have been discovered in many fishes. Particularly, there are 5 types of antifreeze protein, including the antifreeze glycoprotein and antifreeze type 1 to antifreeze type 4. Fishes. Plants also have been reported to have antifreeze proteins in them. The antifreeze protein activity in the plants can be due to proteinaceous molecules or could be because of non-proteinaceous molecules. The protein-based activity in the plants is due to AFGP and AFP, while the non-proteinaceous based activity is due to antifreeze glycolipids. Apart from plants, many fungi, beetles, insects, and bacteria have also been reported to have antifreeze proteins in them. There are a variety of applications found in the industry, in different industries, for the antifreeze proteins. In food industry, antifreeze proteins are utilized in the ice creams to preserve their shapes and taste. Antifreeze proteins are utilized in preserving the crops from harsh temperature. They are used in the process of cryopreservation, in which the mammalian cells and tissues and organs of the human beings are preserved. Also, we are aware that every 10 minutes, a patient is added on the list of organ transplantation, but these organs die out within the 8 hours of donation. That is why we are looking forward to utilize antifreeze proteins in the organ preservation method. The prediction of antifreeze proteins is that they are difficult to distinguish from other proteins type. Some of the antifreeze proteins are observed to undergo structural changes at low temperature. The interaction of ice structuring proteins differ from species to species. This diversity can be due to the difference in geometric arrangements of oxygen atoms in ice. Also, the sequence and structures of antifreeze proteins differ significantly, although they belong to similar species. A lot of work has been done in this regard of prediction of antifreeze, but AFP-CKSAAP is considered as a pioneer work to utilize machine learning approach to predict antifreeze protein. Random forest approach was utilized to identify the antifreeze proteins in this work. In the IAFP, the authors utilized N-peptide composition from AFP sequences of various protein species. In another work named as AFP_PSSM, the evolutionary information in the form of PSSM profiles were used to predict the antifreeze proteins. PSEAAC Builder is an algorithm that converts a protein sequence into a digital vector for the purpose of prediction. In AFP-Cool, 4 types of features were incorporated with SVM for prediction of antifreeze proteins. In RAFP-PRED, localized processing were done on the segments of amino acids and dipeptide composition. To successfully address the prediction using the machine learning algorithms, one must know which features should be selected, and also an appropriate classification method is to be selected. For the feature extraction, we used composition of K-spaced amino acid pairs encoding scheme. The application of this scheme can be found in variety of bioinformatics. This encoding scheme is based on the frequency of amino acid pairs that are separated by K number of residues. The figure shows an encoding of protein sequence having 36 residues. By selecting a value of K equal to 8, the resultant feature vector length is 1200. It is evident from the literature that large values of K contributes to better prediction as the number of attributes are increased. The conventional machine learning algorithms are designed to produce adequate performance results by utilizing less number of attributes to avoid computational complexity. However, a well-designed neural network can efficiently deal with the large number of attributes for a nonlinear classification task. Deep neural network can easily learn nonlinear mapping using ReLU and skipped connections. The classifier is a 4-layer architecture, including an input layer, 2 hidden layers, and an output layer. The number of neurons in the input layer is the same as the length of feature vector. The number of neurons in the first hidden layer is 70, and the number of neurons in the second hidden layer are 128. 30% dropout connections are used to connect the input layer with the hidden layer and the 2 hidden layers. This technique is used to drop some of the units arbitrarily to guarantee model robustness, unbiased training, and overfitting is also avoided. We also used residual connections that helps in achieving faster convergence during the training process and to propagate larger gradients. This figure shows the workflow of the proposed algorithm. The dataset is obtained from the PFAM database in the FASTA format. The features are extracted using the CKSAAP scheme as discussed earlier. The training dataset is formed by randomly selecting 300 antifreeze proteins and 300 non-antifreeze proteins. The test data is highly imbalanced, having only 181 antifreeze proteins and 9,193 non-antifreeze proteins. The neural network is trained using the trained dataset, and prediction results are obtained using the test dataset. For the performance evaluation, we used widely used threshold-dependent biostats nomenclatures including sensitivity, specificity, and accuracy. The accuracy measures the correct prediction over all the classes. Therefore, in case of highly balanced data, if a classifier predicts only the negative class, the accuracy measure will still be very high. Therefore, in our study, we consider the Udens index value as a performance measure, which is dependent on both sensitivity and specificity. The ROC curve is plotted that shows how well a classifier can distinguish between the classes. Higher values of the area under the curve, better the model is at predicting positive as positive and negative as negative class. The 95% area under the curve shows efficient performance of the classifier. A comparison of proposed classifier is made with the random forest and support vector machine classifiers. The graph is plotted for increasing values of k and Udens index. It can be observed that highest value of Udens index is achieved for k equals to 8. This shows that important motifs are present at the distance of 8 residues. The comparison results shows that the proposed algorithm yields highest value of the Udens index. Some of the researchers have used imbalanced dataset for training to show superior performances of their classifier. The algorithm tends to get biased for the majority class, but for the comparison purpose, we use 3 times more negative samples than the positive ones. The results show that the proposed algorithm yields best value of Udens index for balanced dataset. In this slide, you can see a comparison of the proposed algorithm with the contemporary work, and it is evident that the proposed algorithm yields best values of Udens index compared to the previous work. In this research, a deep neural network framework based on the notion of composition of k-spaced amino acid pairs is proposed. Detailed analysis of different learning strategies, as discussed earlier, for balanced and imbalanced dataset, they have been explored, and the method yields high values of the parameter of the Udens index. The proposed algorithm performs prediction by harnessing useful information from the extracted features. However, we believe that the dependence on any feature selection method can limit the performance, and in the future, we would like to investigate one-hot encoding scheme of amino acids with recurrent neural networks. Thank you.